Good day, Year 9. Now we start our new topic about cell division and inheritance. We're going to study a lot about genes and how they are passed from parents to their young offspring. We're going to study about how we may come from the same parents but look different or have different characteristics. We're going to study all about our DNA as well. So let's begin. To, First of all, we're going to look into cell division because any cell, if an organism wants to grow, cells must divide, they must multiply. So, first of all, we study about how cells increase in numbers. There are two ways by which cells divide. When a cell divides, the characteristics have to be passed from the first cell to the two new cells. The information is, as we have studied from the beginning of this year, the DNA has all the information inside of it or stored inside of it, and we have our DNA stored in the nucleus of a cell. This is in animal cells and plant cells and all living cells. There must be a DNA which codes for the characteristics of this living thing. The information, as we said, is stored in the nucleus in a long molecule that is called the DNA. There is another name for it. We call it the genome. So the human genome is just another word for the human DNA. So, as we said, before the cell divides, we have to have information for the new cell because without the DNA, a cell cannot make its new proteins. If it wants to make enzymes, if this cell wants to make antibodies, if this cell wants to make hormones that are protein in nature, all of these, the recipe how to make this molecule, the protein molecule, this recipe is stored in the DNA. So to make new cells, we have to make sure that the new copy of a DNA is made, and making a copy of a DNA is called replication. A chromosome. A chromosome is a section of a DNA. The DNA is a very long molecule, and in a cell, it is usually divided into sections, and these are what we usually see under a microscope. People, when they studied the cells, they noticed that, especially around the time of cell division, we were able to see with the help of dyes to stain the cells, we were able to see thread-like structures or stick-like structures that are stained darkly under the cell, inside the cell. And they started calling these structures chromosomes. Chromo means color, zome means body. So chromosomes are colored bodies. These colored bodies make part of the DNA. Together, all of them is our DNA, and they contain genes. We're going to talk about genes in a minute. And these chromosomes are always seen better during cell division. All right. So this is what we usually see. We're going to look at one of these chromosomes. These are actually two chromosomes. I'm going to trace one for you. This is a chromosome. Here we are. And the next one is, the one next to it is another chromosome. These two chromosomes are held together in the middle by the help of a part called centromere. One of our chromosomes come from our male parent, and the other chromosome comes from our female parent. So that's why we have two copies, because this is how our chromosomes came together. One of them came from the male and one of them came from the female. They're held together, as we just said, by the help of a centromere. Together, these two chromosomes, we name them a homologous chromosome. This is a pair of homologous chromosomes. Each chromosome comes from one parent. And if we look under a microscope, this is what we see. These are very tidy chromosomes. We actually get this diagram by cutting around the chromosomes that we see jumbled up in a cell and neatly putting them just like you see now. This diagram is called a karyotype. It doesn't matter much the name, but I want you to notice that each two chromosomes, they look very equal in size. They have the same size. That's again because one of them came from a male parent and the other came from the female parent. So we have in a human cell 23 pairs of chromosomes. So in total we have 46 chromosomes. Let's look at the circle down at the right corner. This last 23rd pair is actually a bit different. In males, a person will have an X chromosome, which is a bit longer, and a Y chromosome, which is a bit shorter. So this is actually a karyotype from a male person. In a female person, we will have two X chromosomes, which both will be big in size. That would be a karyotype of a female person. All right, let's move on. Now we're going to look at different types of cell division. I'm going to start with mitosis. Mitosis is the type of cell division that happens in all our body cells. So if I look at skin cells, when they make more skin cells, if I look at muscle cells, any cells, hair cells, any part of our body, except the cells of that we find inside the testis or inside the ovary. All other cells in our body divide by mitosis. We start with a cell that we usually call a parent cell. It is not because it comes from our parents. No, it's just the cell that uh, originates or originally produces the two new cells. So, looking at the chromosomes, of course, in a human cell, we will have 46 of these chromosomes, but it will be very difficult to look at 46 chromosomes. So now we simplified them by drawing one chromosome that is red in color and another blue chromosome that is blue in color. All right. The first step of mitosis is making a copy of our DNA. We call that DNA replication. So if you look at the cell underneath, now we have two copies of the red chromosome and two copies of the blue chromosome, and they have joined together by the centromere in the middle. After a while, these two chromosomes, chromosomes, they stand in the middle, and there are threads that try to separate them apart, and they actually do separate the red into two chromosomes and the blue into two chromosomes, and the cell membrane starts to divide so that this becomes two separate cells. At the end, we have two cells, one that has one red chromosome and one blue chromosome, and the other has a blue chromosome and a red chromosome. These are called daughter cells. They are called daughter cells because they came after the division of the parent cell. Again, this has nothing to do with having babies. This is just a way to explain how a cell will divide to make two cells that are an exact copy of the cell that we started with. Okay, so the daughter cells, as you notice, they have the same number of chromosomes as the parent cell. So there's one of the red, one of the blue, just like the parent cell. So this causes us to make copies of our cells, which is actually good because we have cells that die every day and we have to replace them with new cells, or else, after a while, all our cells will become dead if we do not replace them by new cells. We also have injuries and we want to replace the dead cells with new cells. So that is why we need mitosis for growth and tissue repair. There is another way of cell division, and that other way we call meiosis. Meiosis is different than mitosis, and I like to remember meiosis with ME, the one that starts the word meiosis. I start to think of, oh, yes, this is me. This is how Ruba or any person came into this world. It is because of meiosis that created the sperm cells and created the egg cells, that one of them from here and one of the egg cells, one of the sperm cells, they joined together to create this new human being. So meiosis only deals with making the gamete cells, making the sex cells, the sperms or the eggs, the pollen grains in the case of flowers, or the eggs also in the case of flowers. Okay, so this is a special way of cell division that helps to produce a special type of cell. Why are the gamete cells special? Can you remember from the previous chapter? Gametes must have half the number of chromosomes. So we cannot use mitosis. Mitosis produces the same number of chromosomes, so we need to have a different way of cell division. So let's look. First of all, the cell will also replicate. The DNA will make a copy of itself. This is what we will see. Two of the red chromosomes and two of the blue chromosomes. Then there's something very peculiar that happens here. Let's look. When they're very next to one another, they start mixing up some of, as if the red chromosome took a bit of the blue and the blue chromosome took a bit of the red. This is called crossing over. It's not very important in year 9. It's just to explain it, it's just like shuffling a deck of cards. When you play cards with your friends, usually you would shuffle so that you make sure that we don't all get the exact numbers just like everyone. So we have different combinations of numbers. The cell divides first so that one of these chromosomes goes to one cell and one goes to another cell. And then it divides again. So this is something different than mitosis. The cell divides again, so we get only one chromosome in each of the daughter cells that are produced. That means that we have half the number of chromosomes as the parent cell, and we have 4 cells produced by meiosis. Unlike mitosis, there are only 2 daughter cells when one cell divides. Okay, now we're going to draw a table. You have the same table in your study guide. You can open it up and fill this with us. Here is the table where we're going to give details, write it neatly, the differences between mitosis and meiosis. And I think this is very important for you to remember. So please have your pencils ready and write this down in the table in your study guide. In mitosis, we want to remember that this type of cell division takes place in all body cells except ovaries and testes, while meiosis is completely the opposite. This takes place only in ovaries and testes. Never meiosis will take place, never anywhere else. We're talking about humans, of course. If we're talking about flowers, then meiosis will happen inside anthers and inside ovaries as well. Okay? Now, how many daughter cells are produced? In mitosis, remember, 2 daughter cells are produced. In meiosis, we actually have 4. So 4 daughter cells are produced in meiosis. In mitosis, the daughter cells have a full set of chromosomes, just like the parent cell. And when a cell has a full set of chromosomes, we call it a diploid cell. If a cell has half the number of chromosomes, as in meiosis, we say that this cell is haploid. Here, only one cell division takes place in mitosis, but in meiosis, we have two cell divisions that happen one after the other. Consecutive. Consecutive means one after the other. In mitosis, there is no crossing over. Remember when the red and the blue chromosomes, they changed part of themselves with one another? This only happens in meiosis. So crossing over does not happen in mitosis, it happens in meiosis. The parent and daughter cells in mitosis are identical. Exactly the same information is present. The same number of chromosomes are present. You cannot tell the difference if I have two cells that are produced by mitosis. Cannot tell the difference between them at all. But in meiosis, the parent and daughter cells are not identical. So this is important, and this tells us why maybe you came from the same mother and the same father as your brother and your sister, but you do not look identical. You do not look identical to your brothers or sisters, and you do not look identical to even one of your parents. You still have differences between you and one of your parents. So maybe you look close to one of them, but you're not identical. Identical means we cannot tell the difference between them, as in the case of identical twins. All right? Now, let's move over. A gene. A gene is a part of our chromosome that will code for a certain characteristic. A chromosome actually can have many genes. It could be up to 100 or 1,000 different genes on the same chromosome. So it's like a page in a recipe book for me. If I open a recipe book for making cakes, مثلا, I might open a page on telling me how to make a vanilla cake. I can also open another page that will tell me how to make a chocolate cake. So a gene is a segment of DNA carrying a recipe, or to say it more scientifically, carrying a code for making a certain molecule. And actually, the molecules that the DNA tell us to make are always protein molecules. And of course, if I can make a certain protein molecule, such as an enzyme, then this enzyme can help me to make any other type of molecule that I need. It will join different molecules together, maybe to make carbohydrates or fats or whatever other molecule that my body needs. So please remember to memorize what a definition of a gene is. It is a segment or a part of a chromosome or a part of a DNA carrying a code for a certain characteristic or for a certain protein molecule. Okay? Now remember, we can have varieties of the same characteristic. I can have blue eyes or brown eyes. I can have brown hair or blonde hair. And so on and so forth. For each and every characteristic, there are many variations. And an allele, this new word, an allele is one of different forms of a gene. So there could be many different forms of a gene, just like I have just told you. I could have instructions for making a vanilla cake, or I could have instructions for making a chocolate cake. Remember, we just took a while ago that we have two different chromosomes. One we inherit from the male, our father, the male parent, and one we inherit from the female parent, our mother. So, what if we have two genes, right? And here we're going to talk about flowers and petals. What if I have two different genes for petal color for a certain flower? One codes for making white petals and one codes for making red petals. Let's imagine that these alleles are here. One of them, I colored it red. Of course, there is no red color inside our chromosomes, but just to make it easier for you to understand. So on one side, maybe coming from the male part, there are instructions for making red flowers or red petals, and on the other, we have instructions for making white flowers. Now, what would the flower have? Red petals or white petals? Or a combination of both? Usually, one characteristic is stronger than the other. And we're going to talk about that in a minute. Okay? So, if each chromosome holds one allele, if these two alleles they code for the same characteristic. So if these two were red, for example, this organism, we're going to call it homozygous for petal color. For example, here, let's say this plant on a different part on its chromosome, there are codes that tell it to become tall plants. And the code on this chromosome on the right side is also the same as the code on the left chromosome. So if a organism has two alleles and these two alleles have exactly the same information to making the same characteristic, then we say that this is a homozygous organism for this characteristic. Homo means similar. Zygous comes from the word zygote. Do you remember zygote is a cell that is made by the joining of a male and female gamete? Remember? So homozygous means a cell that has the same characteristics uh, in the chromosome. But if they're different, like the red and white petals, then we're going to call this organism heterozygous. Okay, it is heterozygous for petal color and it is homozygous for tallness. Okay, as we said before, some alleles are stronger than others. We're going to call the strong alleles dominant. And because we want to write a code for the allele, we're going to write a capital letter for the dominant characteristic and a small letter. It is going to be the same letter, but small letter for the not dominant or the recessive alleles. So let's say that the red colored petals are dominant and the tall plants, if a plant is tall, it is tall because it has a dominant gene. So we're going to choose R for red colored petals and capital T for tall plants. Okay, so if we have weaker alleles, we're going to give them the same letter but a small letter. So small r is going to code for white petals and small t is going to code for short plants. A dominant allele is an allele that shows its character in homozygous or heterozygous pairs. So, for example, I know the words are too big. Let's take it slowly. Homozygous pair is when the two alleles are exactly the same. So if this plant has a capital R, definitely the petals are going to be red. But if it was a heterozygous plant, it had a capital R and a small r. So what would that code for? Will it have red or white petals? It will have red petals. So that's because it has a dominant allele. A dominant allele shows its character. This is what we are going to see. The flowers are going to be that certain color, red, in either homozygous or heterozygous individuals. Okay? Recessive, on the other hand, is an allele that shows its character only in homozygous pairs. So if I have a small r, I can only see that this petal is white if there are two r's, small r and small r. This is when we will see white petals. If a plant has one of these pairs, let's think again. I'm going to ask you again. If I have either capital R, or capital R, or small r, what petal colors will it have? Let's think together. Capital R, capital R. What do you think? The petals, because they are too dominant and we have no other instructions. They all tell us to make red petals. So what other choice do we have? Yes, this flower will have red petals. Another flower has two different codes, two different alleles. One is a capital R that tells it to make red petals, and another is a small r that tells it to make white petals. Which will it be? Because we know that R capital is dominant, then this flower will be, yes, it will have red petals. Finally, if we have two small r's, two recessive alleles, what will this flower have? What color petals will it have? It will have white petal colors. Good. So this is just a practice for how the alleles affect the overall shape or overall characteristic. Okay, let's move on. When did it all start? It all started when a monk who lived in a monastery, his name was Gregor Mendel, and he loved to grow his own plants, as many people do in monasteries. He used to grow pea plants, and he started to experiment. He had some pea plants that were tall and some other pea plants that were short. He noticed when he studied his pea plants that 75% or around 75% of the plants were tall, while 25 of them were short. Now I want you to think, which of these characteristics is dominant? Is it the tall or the short one? So, did you think? Well, usually, if something is more prevalent in numbers, then it is the most dominant characteristic. And yes, tall tea plants had a dominant characteristic than short pea plants. Now, let's think. If a plant was short, let's give it a letter. Let's choose letter T. If it was a short plant, which pair of alleles would it have? Remember, short is recessive. We don't have so many of them. It's a recessive characteristic. So, would it have a dominant allele or a recessive allele? Should we write it capital or should we write them in small letters? Yes, we should write them in small letters, so it's a small t that codes for short plants. What if a plant was tall? Which pair of alleles would it have? Remember, it could be that both alleles are the same, a homozygous pair of alleles. Or is there another choice? Yes, it could be capital T, or a capital T and a small t. And in both cases, you could never tell the difference. They will both be plants that are tall. All right? This is a lovely game that I like to play when we're together in class. The human earlobe shows us one of the different characteristics that are actually inherited. An earlobe, for those who don't understand what we are talking about, is usually the part that a girl will have an earring sticking to it. Okay? So this is what we call an earlobe. And here I have two diagrams. One with a free earlobe, and the one on the right, it has a fixed earlobe. It's as if it is part of the head. It's not free. Okay? So humans can have either free or fixed earlobes. You can do this at home with your family members and try to count how many free and how many fixed earlobes do we have at home or in class. And then let's think which is dominant. I'm actually going to leave this as a homework for you. But let's move a little together and then I'll leave the rest to you. You're going, after studying a big sample of humans, so try to make this with at least 10 people. You can ask your aunt or your uncle, your grandma or your grandpa to join in just by taking a photo of their earlobes and showing the photo to you, for example, if you cannot see them today. Just a way of counting in a big number of people, how many people have free earlobes and how many have fixed earlobes. And then you're going to decide which of these characteristics is more dominant. And when it's dominant, again, it is seen in greater numbers in the population. So if we have 10 people, then probably 6 of them have the dominant characteristic, and only 2, 3, 4 have the recessive characteristic. Okay? So you find this out for me. Now, you're going to choose a letter to represent the dominant allele. Maybe you're going to choose letter E. Will it be a capital E or a small e? It's a dominant characteristic, remember? So maybe you're going to choose a capital E. And to choose a letter for a recessive allele, you're going to choose the same letter but in a small lowercase. Okay, so it's going to be a small e. And then you're going to look at your own ear and decide which do you think alleles do you have? Do you have 2 of the dominant copies, or 2 of the recessive copies, or 1 dominant and another recessive? We're going to try to find this out as a homework. We're going to study 2 words. The word genotype is the type of alleles that an organism has. So maybe I have 2 alleles that are dominant, so my genotype is going to be a capital E and a capital E. Maybe I have a recessive characteristic, so my 2 alleles are going to be small e, small e. Okay, so this is the genotype, the type of alleles that an organism have. The phenotype is the observed characteristic that is influenced by the genes. So if I have capital E, then my earlobes should look a certain way. I'm not going to give out the answer. It might be fixed or free. You're going to find the answer for us, right? Remember? So the phenotype, maybe my phenotype, let's talk something else, not the earlobes. Let's talk about my phenotype is blue eyes or brown eyes. This is what we mean when we say phenotype. It is the observed characteristic, okay? Here we're going to draw genetic diagrams. A genetic diagram is a way to represent how genes are carried from parents to offspring. And there are many different ways. Let's begin by taking this example. We're going to look at 2 parents, and these are flowers. We have one flower with red petals and another flower with white petals. And let's assume that the red is dominant and the white is recessive. What if these flowers have homozygous characteristics, or they're homozygous for petal color? So homozygous means it has two similar alleles. So this flower, the red one, should have capital R, capital R. So that's the genotype of this red flower. What would the genotype for the white flower be? We just said white is going to be recessive, so it has to be small r, small r. To represent the gametes, we like to draw circles, okay? And the circle is going to represent the cell that carries one of these genotypes, one of these genes, okay? So let's say one of these circles will have a capital R and the other circle will have the other capital R. Remember, the gametes are haploid cells. They carry only half of the genes that we have, or half the chromosomes that we have. So half of them will be in this cell and half of them will be in another cell. Okay? And if we're talking about the white flower, it's going to be the same idea. One small r in this cell and one small r in the other cell. Now we're going to see what could happen. We could have one of these, let's say one of them is a sperm cell, or actually these are flowers. It's correct to say one of them is a pollen grain and is going to fertilize an egg cell. So this capital R is going to meet with that small r, giving us this genotype, R capital R small. It could only also be this way. This capital R is going to fertilize that small r. Again, the genotype that we're going to have is capital R and small r. And the same thing, capital R and small r when these two meet, and when these two meet and fertilize, also the result will be a capital R and small r. All the offspring have the same exact genes. And what will their phenotype now be? Will they have red petals? Will they have white petals? Remember, the red is dominant, so it will win. It will tell the flower to make red petals. So we're going to have red, 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 and red. So all the offspring will be red in color. Oh, that would be really peculiar. Maybe that's what caught Gregor Mendel's attention when he started to play with the pollen grains and take some of his red flowers and pollinate some of his white flowers. He noticed that all the seeds that he collected grew to become plants with red flowers. Okay? And the ratio is we're going to count how many of these and how many of those and put them in a ratio. So there is only 100% of red and 0% of white. When we write the ratio, it is 1 to 0. 1 red and 0 white. Okay? The second scenario, again, genetic diagrams, we're going to look at a different way where we have a red flower again and a white flower. But what if the red flower was not homozygous? What if it was heterozygous? Remember, hetero means different. So there are different alleles for the red flower. It has a dominant and a recessive allele together. So it's a R capital and R small. And this white flower, it's a recessive characteristic. It has to be only small r and small r. The gametes, again, we split them up. So the capital R goes in one circle, the small r goes into another circle. And with the white flower, the same idea. One small r into one circle and another small r into the other circle. And then let's see what would happen if this capital R fertilizes this small r. What offspring genotype will we have? Capital R, small r. And the other probability, what if this small r meet with this small r? Two small r's. What if this capital R meets with a small r, capital and small r, And what finally would we have when small r fertilizes a small r? We get two small r's. Now, what would the phenotype be? We have differences now. We have some flowers that will be red and some flowers that will be white. We will have one red, one white, one red, one white. So what is our ratio? It is exactly 50%. Two chances of getting red and two chances of getting white. So it's an equal chance of getting either white or red. So you can write it in a percentage. You could say 50% red, 50% white, or you could write it in this form of a ratio. It's according to what the question is asking us to do. If they ask us to write a ratio, then we should write 1 to 1. And if they ask us to write percentage, then we'll say 50% red, 50% white. Okay? One final scenario. What if we cross two flowers that are red in color and they are both heterozygous, meaning that they have one dominant allele and one recessive allele? Capital R, small r, capital R, small r. Let's split the alleles, one into each of the gamete circles, just like this, and the same way over there. And let's think of the different probabilities. Maybe the capital R will fertilize the egg cell with a capital R, so we get the offspring will have two capital R's in their chromosomes. It could be that the small r meets up with the capital R, and we always write the capital before the small, so it will always be capital R first and then small r next to it. The other example or probability, capital R with small r, so capital R and small r, and finally a small r and another small r, and this is the other offspring. So the phenotype here, what do you think? Do we have any white flowers? Do we have red flowers? We actually have 3 red flowers and only 1 white. That's very peculiar. Again, did you notice when sometimes maybe two parents with brown eyes, they have a child with blue eyes? And they say, he gets it after his grandmother or his grandfather. This is exactly what would happen when you cross two red flowers. There is a chance that they have seeds that grow into plants that will have white flowers. How did red flowers give offspring to white flowers? That's because we have this situation where they are both homozygous. I'm sorry, when they are both heterozygous flowers. Okay, heterozygous meaning they have two different characteristics. Okay, two different alleles. Here the ratio is 3 to 1. Or if you want to write it in percentage, then there is a 75% chance that the offspring will become red in color and only 25% chance that they will be white in color. Okay? Now, we're going to look at a different way of drawing genetic diagrams. This is a bit more simple, but we need to know both different ways. This is called a Punnet square. And when we draw a Punnet square, we draw a table just like this one, where the parent genotypes go outside the table. So we write, for example, I have capital R for flowers, red in color, and a parent genotype that is small r for a flower that is white in color. We're going to split the alleles, the capital R's, into these two different cells, and the other small r's are going to be split into these two cells. And now we're going to look into each box. What if this capital R meet with one of the small r's? Let's look at the box on the left hand side, the upper left hand side. And again, the same. If another R, capital R, meets with the small r. So let's look at this row. What will we get? These are the options that we get. And if we look again to the boxes underneath, we just look at the box on the top. We have a capital R on the top, we have a small r on the left. So when this one meets that one, we get capital R, small r, capital R, small r. It's a Punnet square. It's much more simpler. It takes less time to draw, but we need to know both ways of representing the genetic diagrams. The inheritance of gender. Remember, we said males have different chromosomes than females. Let's look at a Punnett square that explains how gender is inherited. So, a male person will give either sperms that have X chromosomes or sperms that have Y chromosomes. So, males, they have sperms, and the sperms are different. Some of them will have X chromosomes, some of them will have a Y chromosome. In females, there is no choice. We females have only X chromosomes. So, all the eggs of any female in the world will only have the X chromosome. So, we have two choices of X chromosomes. So, what if the X from the male meets up with the X from the female? What will this organism become? Will he become a male or female child? It will be a she. She will be a girl. XX. And what if the X meets with the other X? Also a girl, XX. So these are the chances that produce a girl or a female. What if the Y sperm fertilizes an egg with an X inside of it? This will grow into a child who is a male, XY. And again, what if the Y fertilizes another X? It will also be XY. So, what is the chance that a pregnant lady gives birth to a male child? Let's look. We have two chances for males and two chances for females. So it's an exactly equal chance. So it's a 50% chance each time a lady is pregnant. If we want to think how likely it is that the child is a male or how likely it is that the child is a female. It's an equal chance. It's a 50% chance that this baby will be a boy and an exactly another 50% chance that this baby will be a girl. So it's fair and square, isn't it? Yes, it's very fair. Which parent now is responsible for the child being male or female? Is it the mother, the cause of the baby becoming male or female, or is it the father? The chromosomes of the father are the ones responsible for giving us a baby who's a girl or a boy. So we cannot blame the mother that she only gives birth to girls. No, or gives only birth to boys. It's not her fault. It's the fault of the sperms that were good enough and strong enough and quick enough to fertilize the eggs. So it's up to chance only. All right? So now we are done. We now have a homework for you. Remember the earlobe homework? You're going to go look for your family, look at your family's ears. Yes, it will be a very funny homework to do, isn't it? And you're going to check if the earlobe is a fixed earlobe or a free earlobe. Have a table neatly done in a copybook and start counting how many of your family members have free earlobes and how many have fixed earlobes. And then try to determine which of these characteristics is dominant and which is recessive, and then try to find out uh, why did you inherit this certain earlobe. Is it because your parents are both dominant or both recessive, or what do you think? We're going to discuss it together when we meet next time, hopefully, and we'll see what did you get, what results did you get. Have a lovely day. I hope you understood everything. If you have any questions, as usual, when we meet together or on my Facebook page, my Messenger Facebook, you can send me any questions you have. Have a lovely day. Goodbye.